Hello,
Hello.
how are you?
Much better.
Oh, good. That's good.
Okay. Bradley, got
Yeah,
a question.
okay.
Uh, that is, use GitHub for depositing software.
Yeah, we do that.
Okay. Are there some idiot-proof instructions?
Yeah, we could do that. I have some tutorials, but it really have to be like, I'd probably have to walk you through setting things up. What were you interested in doing?
Well, I'm working with a kid who's using Python, and I'd like to see him document his code and put it in GitHub.
Okay, yeah, that would be good. I can help with that if I don't know how the best
Okay.
way to do that would be, but
I'll I'll let him know. But if you have a document, you can start with that.
okay, yeah. Yeah, okay, I can set them up with some things, yeah.
Okay, great. I
Perfect.
talked to him at noon today.
Oh, okay. Well, that's great. Okay.
Okay.
Yeah.
We're doing something very strange, and you might want a presentation on it when we're ready in a week or two, I guess. And that is, we're trying to reconstruct pictures from a subset of lines across them.
Oh, okay.
Okay, and apparently, I don't know any. I don't think it's been done.
Right.
Okay, so we've come up with a very naive in-painting algorithm, and it seems to be working.
Oh, good. Now, this is the one for the breast cancer.
Yeah, it's leading up to it. We're not doing the breast cancer case yet. Just a standard picture,
All right.
grayscale.
All right. Yeah, that sounds great. Yeah, I'd like to see it when it's done.
Yeah,
Could do a
yeah. It's
presentation of
we're about half done.
it. Okay.
Okay.
Yeah. Great. Morgan, how are you? Usually
We
he's.
never see it. We only
Oh
see
yeah,
it
usually he's running errands or something in the morning. Good morning. Yeah, the morning school run. So that's great. Yeah, so that's I don't know if Susan's gonna make it today, but sort of our nucleus, no pun intended. Uh, yeah. So I've got some things today. I know Morgan wanted to go over this paper, this hypergraphs paper that I have out. Probably also talk about some things. Okay, yeah, and then Morgan says, been meaning to follow up with Susan and Morpho. Oh, the Morpho program, yeah. Okay. So let me share my screen. All right, so I'll start with the hypergraphs paper, I guess. So recently, this is put on the arXiv. This is something I'm working on for a while. This is this paper, Hypergraphs Demonstrate Anastomoses During Divergent Integration. And we talked about this in a meeting, several meetings last year, I think. And so it's just been kind of going along, and I kind of put some finishing touches on it and put it out. So basically, what this paper does is it uses hypergraphs or graphs, basically graphs within graphs, to model morphogenesis, embryogenesis, etc. And so, there's this work that has been done earlier. This is on different cell differentiation processes of spatial networks. So these are embryo networks that where you take the embryo and you put it into a network representation. But that's what we call a first order network. So a first order network is where you have this point cloud of cells. This is, of course, if we take microscopy images and we extract the centroid out of each one, and then we plot them in this 3D spatial coordinate system, or maybe even a 4D coordinate system if you go over time, and then you put together, you find the connections via a number of criterion. So it could be the distance between the cells, it could be signaling molecules, it could be other things. So this point cloud here, as you can see, this point cloud is growing as we go through time, and we get cells dividing and even differentiating in space, but then also in terms of fate. And you can build all sorts of networks. So you get these point clouds that grow in density and size over time. Then you get, you can like investigate what they look like, sort of in two-dimensional slices. And then you can actually build these circular networks. So these networks are rather disembodied, these plots, because what they do is they just take, they put things in a circle and they show the connections between cells. These are kind of ordered by, the anterior end and the posterior end. But you see this connectivity in the middle, all these lines. And those lines kind of represent connections from, the anterior to the posterior side, or the anterior to another cell in the anterior portion of the embryo, and so forth. So you can see that in this case, you have some patterns of connectivity. You actually have to run graph statistics, network statistics, to really kind of get at some of the properties of these networks. The other problem, of course, then, is that those descriptive parameters oftentimes don't describe very much about the network. They just describe the global parameters. So, for example, there are different parameters like centrality and other types of parameters that give you basic information about the network as a global entity, but not local entities. And so there are a number of network approaches like community finding and things like that, which we actually do, I think, later in this paper. We'll look at for communities in these networks. But those are other exercises you can do, and it helps you describe the network a little bit more in detail in terms of the local components of these networks. So you can see that this kind of connectivity is different than this one. You use different connectivity thresholds. So you take two cells pairwise, and you use your criterion, and you set a threshold for connectivity. So you can have a very permissive threshold, for example, and it can be, you can have more connectivity. You can have a more restrictive threshold, and you have less connectivity. And so as you drop that connectivity, or you bring that connectivity threshold up, you start to get these sparser networks, but you also start to reveal some structure in the network. So you see here, this network here has basically, this is it's not a random network. I've done tests with random networks, and those are different than this. So there are these types of network topologies that are non-random. There's scale-free topologies, small world topologies. This is more of a scale-free topology. It's not random, but this one is more scale-free than this one, I guess, is the lesson. And you end up with these clusters, which reveal patterns within, like, locally in the embryo. So you can see that there are certain parts of the, this is actually where you get down the lineage tree a bit, and you start to get places in the P1 hemisphere, the posterior end of the worm embryo, that have these sort of clusters of cells. So that's what we were doing in this paper, and there are different tests you can do on this. You can get degree, network diameter, graph density, modularity. You can actually look at the number of communities, and then the clustering coefficient. So those are all globally descriptive parameters. And then you can look at that global network, and you can say things about the global structure, and you can get these summary statistics. but they don't really tell you much about the the network structure other than just this snapshot global description. So you can look at these network statistics over time from the 16 cell to the 128 cell, and you can get some sort of, maybe like a time series plot over the number of as the number of cells increases through division and their differentiation events going on. This structure of the network changes. These statistics will change as well. So the degree will change, the network diameter will change. You can see that as you increase the threshold, you get these large gains in network diameter. So as the threshold increases and you get this more restrictive criterion, you saw in, let me zoom in, you saw in the figures sort of intuitively that you start to get these clusters, and that represents a larger network diameter. So that means is that if I start at any one edge of the embryo and I want to go to another edge of the embryo, it takes more hops across different cells to get to the other side. And so as you increase the threshold, in this case, as it goes up and you're being more restrictive, then you get a larger network diameter. And there's this big jump at certain points. You can see that in the 16 cell embryo, for example, the network diameter doesn't exceed 8, and that would be obvious because there are fewer cells. But when you get to 64 or 128, when you get to the 0.95 threshold, you really start to get large network diameters. And so these are good, descriptive statistics, but they don't really get at the sort of the properties of the lineage tree directly or some of the local features. And so, there are some other experiments in here, like the clique analysis, which is where, you look for areas that are densely interconnected. So, a lot of people look for cliques, and, they'll do other things like community finding to find these local substructured groups in the network. So this is a clique analysis, for I think the 128 cell embryo. And, it kind of tells you a little bit about, these sublineages in the lineage tree. So when we have our AB and our P1, which is the anterior and the posterior end, you start to get these sublineages, which are based on divisions of AB, divisions of P1, and you end up with 8 founder cells, what they call founder cells. And those are 8 sublineages that continue to the hatch and the post-embryonic period. What we can do is we can take those subnetworks and then analyze them and see if they match cliques. And, that's what this was supposed to be. So this paper was in 2018. And I bring it up because it kind of motivates what I'm doing in this other paper. So I've been working on this idea of hypergraphs for a while. And the idea behind hypergraphs for this type of network is kind of to take different, take the lineage tree and represent the cells in these sublineages as not only the individual cells and their connectivity, but larger categories that are connected, not just in space like we did with this network, but also in time. So if you go to the YouTube page, we have a channel, or a playlist, they call it, for network science. And so we have a lot of interesting things on the network science channel, or the playlist. We have a couple of presentations that have been given at the Networks Conference and NetSci Conference, a couple of other presentations. But these are the 2 that I wanted to draw attention to for this recent work, and that is most relevant to this recent work. So this is the first one. Hypergraphs demonstrate anastomoses during divergent integration, and embodied hypernetworks, graphs of spatial anatomical embeddings. And so these are kind of go over this work, but in different ways. And there's also this other video, embryo networks, computational development and neural simulation. This one includes some of this work as well, but it's not, it's about more broadly what the network vision is, that includes all of this kind of work. So this is the preprint. So this is hypergraphs demonstrated anastomoses during divergent integration. So an anastomosis is like a pathway or a place where two things are joined together. And that's an anatomical term, and I think Dick mentioned it once in a meeting, and I used it in the title to kind of get at like what some of these graph structures are actually doing in terms of describing the biology. So it's like you have the spatial network, but you also have this temporal component. You have the lineage tree, but you also have the anatomical spatial relationship that is preserved. And then you can describe things like anastomoses or other types of division events. You'll see what it looks like. So this paper kind of goes through, it's very theoretical in the sense that it doesn't use it doesn't use data directly. It does a lot of hypothetical modeling of some of these things, but then there's also some data in here. So there are two. So basically, developed hypergraphs for two or modeled on two organisms. So it wasn't actually taking the data set from the organisms, but it's modeling it on sort of an exemplar organism and then building a model from that. So the first one, of course, was C. elegans, and this is something called a two-airy tree. And then the nostoc punctiforme, which is the cyanobacteria that we talked about in the group. This constitutes multiple discontinuous two-airy trees. So the idea is that these hypergraph model development, and the lineage tree is an n-airy tree, which can model two or more descending categories per division event. So if you have, if you think about a lineage tree as a binary tree, that binary tree then can be, you can represent the tree, and each node has things in it. So let me draw this on a board so you can see what I'm talking about. This works better if I do it this way. So you have a binary tree that has a root and has nodes coming down. And two-airy just means like it has two descendants. So the term n-airy is just how many descendants does it have. And so this is a term from mathematics, but basically it's where, you have a single cell divides into two cells. But that's not all we're interested in, of course. We're interested in the relationship, the ancestral descendant relationship. So it's ordered. And then if we open up one of these nodes, we see it's not just a node, it's a category. And inside the category, there are things inside of it. And in fact, there's a network inside of it, if we would like. We could have a network in there, things that are connected, or they don't have to be connected. The point is that in each one of these nodes, you have things. So you could actually model this as single cells. You could model this as categories of sub lineages, whatever. There are constituents within each of these nodes. They could be cells. They could be molecular events. They could be things inside the cell itself. So there are a lot of properties that you can model within each of these nodes. So it's a hypergraph. So it means that there's a graph here. There's a graph structure, but there's also this structure that exists within each node, and those are connected. So a lot of times with hypergraphs, you'll have like a graph within a graph, and you'll have you'll have nodes within a single node, and it passes things on to a descendant. So we can actually take the constituents of this node here and move it, pass it down to this node here. There's some sort of inheritance rule. Now, not everything in this node is going to be inherited by this node. We can have division events, so we can have a doubling of the things inside this node. We can have things, we can lose constituents. We can, do whatever. Now, in the case, what I'm going to show you, it isn't just, a direct, what they call a DAG or a directed acyclic graph. So a DAG is a directed acyclic graph, and that represents a lineage tree. Oftentimes, they'll use this terminology just to say that everything goes from the root downward and that there are no crossings or intersections between nodes that kind of divide out. So you're always going outward in branching, doing subsequent branching phases, but you never actually join back together. There's no ramification. But in this case, we're going to ramify things. And we're going to do that by having, go to the next slide here. We're going to do that by having like different categories that come down like this. And then we'll kind of, I want to do this kind of just to show, you'll have crossings into different categories. So if you think about this as like a stem cell, this is a neuronal lineage. This is like a, muscle. The muscle cells will descend from this category to this category. You might have a doubling event. In the neurons, you might have a doubling event. And, the idea is that you have roughly twice as many muscle cells in this node as you do in this node, and twice as many neurons in this node that you do in this node. So you see you have this division event, or maybe this differentiation event here, and you have this proliferation event here, which is division. And then, that's one round of cell division. But it doesn't mean that all the cells maintain their identity. So one of the reasons why we want to use hypergraphs is to, kind of allow for cells to switch maybe from a neural precursor cell to a muscle cell, or a muscle precursor to a neuron cell. Or what's more likely in some of these graphs is that you have some early, some cell that's like basically a developmental cell or a stem cell that switches into a new fate. So you have cells that this node, the stem cell node may double. So you might have something like this, where you have a stem cell category, and then you have stem times 2, and then you have muscle, and then muscle times 2, and then this is stem times 4, and then you'll have maybe something from this category move down into the muscle category. And so when we sum up all the cells within this node, it will be all of the muscle cells here. So say we have 16, this will be 40. We take the 32, we take this 16, we double it, and then we have 8 cells that migrate from the stem category into the muscle category. And then this stem category will, say, be here 8, 16, and then this is not 32, it's a subtraction of that, so it would be 24. So this would actually not result in a doubling, this would be times 2, I'm sorry. So it would now result in a doubling, but it would be, doubling minus 8, because 8 cells have moved into this category. So that's the way these hypergraphs are set up. They're set up to allow for these kind of changes in category, and that's the way that we kind of represent changes in differentiation and changes in fate, or changes in the cell state. And so you can actually go down beneath or below sort of cells within cells. You can have cells, like the nodes can represent cells, and then inside it could represent molecular components. So it would be a little bit different setup there. The molecular components, maybe transcript counts or something like that, would change in a similar way, but it would be a little bit different setup. But it allows you, it's very flexible and allows you to represent these different events and things like that. So one of the concepts that I kind of started out with, and this was during the Networks conference in 2021, I gave a talk on divergent integration. And divergent integration is the idea that you take an embryo that's differentiated and it starts to develop, you start to have these differentiation events, and into tissues, and those tissues diverge in terms of their connectivity, but they retain these integral sort of connections. So, for example, if you have, like in a C. elegans embryo, at some point you begin to have a nervous system or a connectome, and so in the connectome you have, like, it looks like you're kind of in this stage of the embryo still. You start to get neuronal cells up here, some neuronal cells down here, and then you get the rest of the cells in the background. And so they can be of different fates, but they're kind of disconnected functionally from these neural precursor cells as they start to differentiate into, a nerve and into a connectome. And so these connectome cells are connected in different ways, and they're distinct from the rest of the cells in the embryo. So they start to diverge in terms of their function, and there's and what they're kind of related to. So they start to cluster together, but they still have these connections to other cells. The connectome in the head and the connectome in the tail are weakly connected. They're going to be more strongly connected later in development. But you also have these other cells in the embryo that are still connected to the connectome, obviously, because they're going to form this unified organism. But they're going to have weaker connections than the cells in its own category, which might be muscle cells or other types of cells. Okay, so back to this paper. So that's divergent integration. And it's going to play a role in these networks. It's going to play a role in sort of this ramification in the network. So I'm going to get right to an example of a hyper network here, or hypergraph. So this is the category. So I've used category theory to represent the different categories. You can have two categories here that come from an initial category. You have X and Y, and you can have interchanges between X and Y. You can have interchanges between X and Y in the next generation, and so forth. This is an example of a hypergraph. Now this is modeled not on C. elegans proper, but on sort of an idealized form of C. elegans. So this is the hypergraph model here. And so you can see that it starts with the one cell, and you start to get these doubling events throughout the lineage tree. And it doesn't look like a conventional lineage tree because it's tracking these categories. And so this gray category is what we call developmental cells. These are like the stem cells that you see in C. elegans that are undifferentiated. You start to get the germline that diverges early. So that's divergence. The germline is less integrated than the other types of cells will be, but it's still part of that organism. And so it's still part of this tree. You're not going to see any ramifications from the germline. And so, that's something to keep in mind when we see some of these other categories. So we have the germline diverge early. Then we have start to get some other things that diverge. We have this connectome and these white net nodes that have, you get these small number of neurons that initially diverge, and then you get more of these like these developmental cells that form this embryo network that start to sort of add to the connectome. As cells and developmental cells differentiate into connectome cells, they move from one category to another. So now they're part of this network within a node. And so they're forming this connectome. You start to get, in time, you get these categories that grow in size, and they exchange with other categories in time and other categories in function. So you can see that it gets pretty messy towards the top of this, where you have arrows going in all directions. But that's just simply because you have more categories where things are being interchanged and things are, moving forward in time. So that's a very different representation of the lineage tree than what we maybe see in the Solston lineage tree, for example. And this is not C. elegans, but it's a model based on C. elegans. And the reason I did that was because I didn't want to have to, I just wanted to kind of show a theoretical model where it's very clean. So this, you can see, for example, you can define things like the 8-cell embryo or the 32-cell embryo. In this case, in the 32-cell embryo, you have 28 developmental cells and 4 germline cells. In the 8-cell embryo, you have 7 like the founder cells, the developmental cells, and then the one germline cell or germline founder cell. And so you can go up and you start to get like where, you have to define like different stages of the embryo, and you have more and more categories to consider. So it gets a little messier as you move upward in time. So that's for C. elegans. Now I go through a little bit more about this, about describing the hypergraph, spatial hypergraphs. And so this is a spatial hypergraph. So this is actually a little bit different formulation. This is where developmental time is now going downward. Reasons that we want to be able to show these things kind of unfolding in time. So this is the 128 cell embryo, and this is the 256 cell embryo. Again, it's not C. elegans, it's an analog of C. elegans, but this basically shows how these kind of categories that we saw in the last figure translate into spatial distributions. So this is the anterior posterior axis at the top, and this is the left-right axis here, and then we have our 128 cell diagram here and the 256 down here. So we have these connections between the doubling event. So we go from our large category of developmental cells at the 128 cell stage, and we move down to the 256 cell stage where we retain some of our developmental cells. We also get different categories of germ cell. We get these other differentiated tissues. And so it's the 256 cell embryo in this diagram is much more diverse in terms of the categories. So we have different categories that are smaller or larger than what we see up here. So there's an accounting here where you say, basically, if there's a doubling event, you have doubling, but you also have contributions to new categories, contributions to different categories, and the like. And so within each of these nodes, you get a network, and that network can be different cells that are interacting. And so you can structure the network in that way, but you can also have a representation where you have these categories that are also interacting in a network. And this can be done spatially as well as just simply temporally in terms of that lineage tree. Okay. This is actually a different example. This is the cyanobacteria Nostoc punctiforme. And so this is where you have a bunch of cells in a chain. And this was another paper we discussed in the group where, they basically have this very defined set of rules for morphogenesis, where you have cells in the middle that divide, and they have different factors in them, and they form different functional categories. So here you go in time, you have this chain that's growing, and so there's this phenomenon called restricted heterocell differentiation, where these this chain will grow by cells in the middle of the chain dividing and increasing the length of the chain. So here we have 1 cell, 2 cells, 4 cells, and 8 cell categories. So as this cell starts to divide, it starts to contribute to this category. And so you have these networks that are discontinuous. You have these discontinuous trees. And so they kind of go through the chain, and you can visualize how this chain grows using this kind of an approach. You have the hypergraph, the cells within that hypergraph, and the cells within there, they have molecular components, and you can model those as a network. So that's why you might want to do something like this. It isn't in the same way that we see in the C. elegans example, but I think it provides a different perspective on this kind of methodology. So it's basically the same type of thing. It's just a little bit different topology. These are some graphs to show like the sort of the expected output that we might expect from one of these hypergraphs. So if we have a number of cells or a number of molecular components within the hypernodes, those hypernodes will have a distribution. So, all the components of those hypernodes, you can take a spectrum and read it out. So each hypernode, which were these colored nodes, will have some sort of output. It's not just the network that interacts and changes its shape and its size, over time, it actually can output different types of things that are in it. And so you can get a spectrum of cell types. You can get a spectrum of, cell types are different in different conditions. And then you can, so this is a spectrum where you have different types of cells, a very small spectrum, but you can get all sorts of spectra that have different distributions. And this is counted in the number of cells. So it's basically a histogram that tells you it's a description of that network or that little subnetwork. And so this is another example where you have transcriptional regulators of protein types. You have different types of proteins, and this is more of a spectrum where this is based on some pseudo data that was generated. So this is more of the distributional character of it, where you get these uneven distributions of different protein counts within one of those hypernodes. And so it's descriptive of the structure of the network, but it's also descriptive of what's going on inside the hypernode. What's the process? And so finally, you can actually do this for like a connectome where you have this output behavior. So you can have these connectomes that link from the sensor to the effector. And you as you have like your sensor, which are your sensory organs, and your effectors, which are like your hands or your legs, we can think of like an insect. And this is a good example because in insects, you have a relatively simple mapping between the sensory organs and the effectors. And you think about the things in between the connectome, and that connectome will grow in a way that's similar to what we're trying to do here. So we have, in development, we have this connectome that grows in size in terms of the cells in it, different categories of cell. And so you can actually account for this, this sort of developmental growth in the connectome that's embodied in an organism in this way as well. So I showed a spatial hypergraph. I showed a temporal hypergraph, which was analogous to a lineage tree. And then now this, which is embodied in an organism going from like the anterior to the posterior end, or the sensor to the effector. And this just models the nervous system in terms of its detail. And so, the different categories of cell, the different numbers of cell that are there, and so forth. And so that's it. There are a lot of nice references in here on hypergraphs, on systems biology, on other types of things. A lot of some category theory in here as well. And yeah, it's really, this is something I've been working on for a couple years. So that's all I have on that. If you want to go to the YouTube channel, I have some talks where they're kind of more condensed, and I kind of give some examples. So we have some, oh yeah, thanks. So we have some questions in the chat here. Morgan, I guess a long time ago, said, I've been in touch with the principles behind the recent Drosophila simulation. Oh, go ahead.
Sorry, this was before you'd even started.
Oh, yeah.
But super interesting. And I just dropped a link in to a paper, a very network science paper on the Drosophila connectome. that again, potentially of interest, just in terms of I'm curious about the latest in network science to identify circuits and then going one step past that, potential logic circuits, and not sure how much of this is relevant or how much of this is embryogenesis, because this certainly this particular network science paper is focusing on an adult brain, right? So definitely differences there in terms of looking at developmental graphs, and one last thing, it's if I haven't already, I think I posted this somewhere, but the Greta seminars. This is like it's graph. Oh, I can't remember now the acronym, but it's basically transformations on graphs seminars, and like how that could be useful, not only in terms of how networks can have multifunctionality, but also just embryogenesis, or how networks are changing over time as an organism grows. But I'd mentioned this before you got started, just because the Cog Lab in Paris has some people interested in this Drosophila connectome flywire that is a kind of crowdsourced. It's all academic labs, but this multi-center repository flywire that is got something like 120,000 proofread neurons at this point, something like that. And what they've been doing with it in terms of some pipelines they've got using ImageJ Fiji to like skeletonize that and then turn it into basically more like connectivity weight matrices, and then use like spiking neural network simulators like Brian 2 in this particular case of Berkeley to do to then look at least some aspects of neuronal modeling with it, right?
Yeah.
And so don't want to get don't want to get you off track of the presentation, but that was like basically all my comments are around that.
Oh yeah, that's that's great. The bioRxiv paper here, is that the paper you were talking about?
Yeah, that's just the one that was very much like network network statistics, I think it was called. Yeah, network statistics, right? And so I more summary measures, but again, like how it's certainly a lot of network science is focused on finding cliques, finding subnetworks, right?
Yeah.
And some of the there's actually a lot of so talking with Shu and the first author of the Brian 2 simulation. There's a lot of great videos from the Flywire Princeton group that just kind of present what's there, as well as the tools that they're using. This Flywire Brain, Fly Brain Lab. So from the Fruit Fly Observatory. This is all new to me, so forgive me if this is old hat, but some really nice resources. And what's great is just that like it's a large complex network that's kind of been curated and you can do stuff with. And I'd love to learn more about how C. elegans and OpenWorm is matches that, or is doing things similar to that.
Yeah, and we haven't really gotten too much into neural modeling for that, but I think the idea is to kind of come up with different representations for networks. So I was looking at this paper here, the network statistics, and they're doing like a lot of different statistics, but so they're trying to find these 2 and 3 network motifs. So it's just like kind of if you look at like 2 nodes that are connected, or like a triangle, which basically a 3-node network motif will form a triangle. That will give you some information about like local things. But this the other thing which they talk about, a population of highly connected neurons known as rich clubs. Those have been well characterized in C. elegans, and getting into like what the development of those rich clubs looks like. That's a really interesting question. It's not something that we've done, or I don't know if we could do it, but maybe with simulations. Like, why do you have rich club neurons? What's the point, or what does it actually do for the network?
And no, go ahead.
Okay. So and then Dick said, please define hypergraph, which was, and then actually Morgan has a description of hypergraph, which is hypergraph is a generalization of a graph in which an edge can join any number of vertices. So basically what happens in a hypergraph, and I didn't go over this formally, but I'll do this. So like I'll compare it to a regular graph, and then I'll show what a hypergraph looks like. So we have, in a regular graph, we'll have like a node here, a single node, and then we'll have maybe a connection to another node, and a connection to another node, and a connection to another node. And this is one of these triangles that I mentioned where you have 3 nodes that connect singularly to its nearest neighbor, and you have this kind of a motif. Now, in a hypergraph, you have like a node here, what they call hypernode. And hypernode just means that it has things within it. So you might have this motif within a hypernode. This would be a single node, but there'd be 3 nodes within it. So now when you move to another hypernode, and you have the same motif, and let's say in this case we're not thinking about like ancestor-descendant relationships. We're just thinking about this kind of connectivity here. So here with this edge, you have 2 nodes that connect it. Each node has a single thing. So there's an exchange of things between these 2 nodes, A and B. And then these nodes here are A and B, but you also have A prime, B prime, and C prime. And so the idea here is that you can transfer things between nodes. These connections, like A prime will be connected to A prime prime down here, and it can also be connected to B prime prime down here. So you have these connections across different networks. So it would be like if you had another network over here, which is like A prime, B prime, C prime, and you have not only these nodes, but then connections between these nodes. So you have this sort of second layer network. And it's like, you're connecting not only within your own clique or this triangle, but you're connecting with another triangle. And those connections are isomorphic, meaning that they can be connected in different ways. So you can connect from A to A prime, B to A prime, and so forth. So it's almost like you have these 2 networks that are sort of in parallel, and they're connecting together. So like basically in this case, you have one node connecting to another node. In the hypernetwork's case, you have one node connecting to another node, but there are many connections that move along this route, and they're connecting to different things. So the connectivity isn't just from like one node to another, it's to various constituents within the node. And so that's the difference between the 2 things. You can have connectivity that's very, very complex, even just having 2 nodes there that are connected. And so it's kind of hard to get your mind around, because it's like, well, I just thought that they were 2 nodes, but you're nesting this stuff, and then you're saying, well, any one of these constituents of node A can connect to any other constituent of node B, and that creates a very complex system of accounting to see which thing goes to what. And it also allows for a lot of these kind of processes in biology where you have, things nested inside of other things to be represented in a way that you can really kind of maybe understand. And so that's what a hypergraph is compared to a regular graph. So any edge can join in any number of vertices. And so in an ordinary graph, an edge connects to exactly 2 vertices. So that's the difference there. And then, yeah, check out the YouTube playlist. And so that's all I wanted to mention about that paper and some of that work. Any questions at this point? So, yeah. Okay. Well, yeah. So let me know if you have any questions or comments. So last week was the Learning on Graphs conference, which is a conference we did last year. We never got around to submitting a paper to it. We were working on the graph neural network stuff last summer and this summer. And Learning on Graphs is basically a graph neural networks conference. But they also kind of venture into this world of graph theory and networks. And so we weren't able to submit a paper this year. Last year I wrote a blog post on the event, and I used it for this year's GSOC project. This year I was able to kind of catch up on sort of the broad outlines of Learning on Graphs, but I haven't delved into any of it. And I want to do a recap of that conference and say something about what's going on there, the major themes. But I haven't had a chance to do it, so that's something I need to do. It was like I have all the materials bookmarked and I'm ready to go, but I didn't get a chance to do it. So we'll see. I'll try to give a recap on it. Maybe I'll do a video, or maybe I'll do something like that too. But it was a really interesting conference from what I saw. A lot of things, especially with respect to topological data analysis, and there was some work on asymmetries in networks, in this case neural networks, that you can use to process data. So graph neural networks are basically taking features out of your data, finding the connections between them, building these graph embeddings, and then using those to process what you've analyzed. Use it as your model. And some people they've been using a host of very innovative techniques to try to get at try to improve performance. One of those being asymmetries, so where the network that you're using has some sort of asymmetrical properties of information processing. People were using topological techniques, to represent geometric graph neural networks, to represent the data input, things like that. So it's really interesting stuff. It's something we can kind of get into. I'll find some good examples maybe and bring them to the group as a whole. So I don't want to overwhelm people with it, because it's there's so much there now. It was several days of talks. So that's kind of okay. Any other questions? Comments?
Just want to catch
Okay.
up on the full week of those talks.
Yeah. Morgan put a couple things in the chat. This Greta package. Yeah.
Well, that's the seminar.
Oh, this is a seminar series. Okay.
Yeah, and so might scroll through there at some point and just have a look and see if anything's interesting. And then the ICTS, was it Winter School on Quantitative Systems Biology has a number of interesting talks. I think I posted one which was the energetics of embryogenesis or embryology.
Okay.
This
is bioenergetics.
But do they have the list if you go down? The schedule.
They have speakers and topics here.
Okay. Yeah, they yeah, and great speakers.
Yeah.
For covering this kind of material. Oh yeah, cell metabolism as an economy, and then self-organization of metabolic networks. Yeah, lots of interesting stuff.
Yeah.
I mean,
a measurement calorimetry. That's interesting stuff. I mean, we did a paper last year where I read through a paper where we talked about like people were doing this in embryos where they were measuring like the energetics of an embryo and the embryogenic process, basically looking at I think they were looking at metabolomics as well. But yeah, it's interesting. People have done this, but it's not in the mainstream really of.
Yeah. But the other thing I just wanted to say was that because I forgot this at the Saturday meeting, but there's a new biolab space in San Francisco that's opened up.
Okay.
Like a community biolab, and anyway, it was someone who's done this before in other cities. So he's got some experience. Hopefully, he's got enough connections to bring a community around to keep the funding going. But they're already getting some support from the city that is nice to see, because they took over a reclaimed, a derelict building. But I was wondering whether like they're gonna have some cold rooms and things like that. I didn't, I was just like, how about cell culture? I knew we had some shared connections between the hackerspace noise bridge and this space, so I pointed them to some particular, like, I thought easy projects. Again, not sure whether they're going to have the facilities, but I was thinking like hopefully they would get a microscope, or at least they'd have a space for a good microscope. And I was wondering about at least doing some what's the geometric structures that form I forget the name of those. Dick, what's it's the single-celled organism that makes a silicon shell?
Oh, diatoms.
Yeah, diatoms. Yeah. Sorry. Yeah. Again, I wasn't sure, if there's anything of interest there, but I certainly would love to have a good space for a microscope to set up and not be disturbed, and or at least have some functionality and some video recording. I'm like always jealous watching these Journey to the Microcosmos videos.
Oh yeah.
Yeah. By the way, there's a technique which needs to be developed for diatoms. That shell is made of silica.
Yeah.
And it's hard to look through. Because its refractive index from water is quite different. So the idea is to add something to water which is non-toxic and matches the refractive index of the silica.
I see.
I call it cloaking of diatoms.
Okay. That's cool. I mean,
and as a hint, I suspect the material that one could use is methyl cellulose.
Okay. I'm going to follow up with you on that because I used to have a grad student working, sort of doing small jobs at the lab, and he was in the optical, like, photonics group at Berkeley, and they were doing this kind of like negative refractive index stuff,
This is a negative. It's just matching the.
No. But he would know good materials, and he would know how to test it and things like that. Like he would be able to point me to people at Berkeley. It might be able to help.
The refractive index is around 1.4 for the diatoms, so it's not. I don't think it's going to be that difficult to match.
Yeah. You don't need full Klingon cloaking.
The point is that there is almost no work which shows what's going on inside a diatom.
Interesting.
And
okay.
Jeremy Pickett Heaps. You should look at his movie.
Okay.
He doesn't do cloaking, but he's done some spectacular imaging.
Okay.
The whole video that he produced is online now with the Russian journal.
Okay.
I can look it up for you if you want.
I'll follow up with you. I gotta get in the car to take this girl to school.
Okay.
But I'll follow up with you, Dick. Thank you so much.
All right.
Okay, sure.
Thanks, Morgan.
Take care.
Yeah, take care.
I'll put my email in the.
Oh, I've got it.
You got it. Oh, okay.
Yeah. Thank you.
Whoops,
Cheers.
whoops.
All right. All right. Yeah. There we go. All right. So yeah, that's it for today, I think.
Okay.
All right. Thanks.
Take care.
Take care.
Bye.
Bye. All right. Now I'd like to talk about a few papers in a category I'm calling New Directions in Morphogens. And so, of course, morphogens are these theoretical constructs that we often talk about when we talk about morphogenesis and the theory of morphogenesis. So let's get started. So this first paper is this is by Amy Shire and Alan Rodriguez and co-authors. And this is a research article summary of a paper we'll get into after I go through the graphical abstract and some of the details outlining this. I'm going to read the introduction and then go through some of the figure. During vertebrate organ morphogenesis, large collectives of cells robustly self-organize to form architectural units, which are bone, villi, or follicles. These forms persist into adulthood. So in the Hypergraphs presentation I gave earlier, we had these categories of cell types, and those persist as, they grow and change, but they persist as categories from some point in development to adulthood. Over the past few decades, mechanisms of organ morphogenesis have been developed predominantly through molecular, genetic, and cellular frameworks. So this is where people look for genes that are expressed, and people look at single genes in cells, and they characterize this in different ways, but they have not linked it very effectively to morphogenesis itself. So this is what a lot of the empirical research has been about, but of course we have all this theory that we need to link to. More recently, there has been a resurgence of interest in collective cell and tissue mechanics during organ formation. So this is where we have this literature. I think we've been reviewing it over the past several months, where you have mechanics and people are looking at different mechanical aspects of tissues. But again, we're not linking all this together theoretically. This approach has amplified the need to clarify unambiguously link events across biological length scales. So different biological length scales are important for not only for understanding how molecular events link to morphology, but also in terms of complexity theory. So for example, we could look at the length scale of an entire embryo, the length scale of a tissue, or the length scale of a couple of neighboring cells interacting with each other. Those are all important to look at sort of the complexity of an embryo or some other collective structure. Doing so may require reassessing canonical models that continue to guide the field. The most recognized model for organ formation centers around morphogens as determinants of gene expression and morphological patterns. So, of course, in Turing morphogenesis, we have this concept of morphogens. It's a theoretical construct that Turing defined as this thing that guides morphogenesis. The question is, what are morphogens and how do they behave if you take any one system and you find particular molecules and you find how their mode of action and things like that. So the classical view of a morphogen is a morphogen gradient specify differential gene expression in a distinct spatial order. So this is where we have these gradients of morphogens. They specify differential gene expression, so we have a gradient of morphogens that are expressed perhaps in a certain region of the embryo. And then that gene expression is differential, meaning that as those gradients overlap, you get stripes or you get boundaries. When you have differential gene expression in one cell type versus another cell type, you get different gradients. Those gradients interact, and ultimately you have different gradients set up in different parts of the embryo. And this is how you get pattern formation. Because morphogen expression co-localizes with emerging feather and hair follicles, and this is where some of their previous work has led to looking at the formation of feather and hair follicles in the epidermal layers of skin. This has served as a paradigmatic example of such morphogen pre-patterning mechanisms. And so this is an example here of what they're trying to do. So they have this is from a previous paper. This is sort of their model system where they have organ morphogenesis. They have this budding, they have the margin and the core of this hair follicle, they have a layer of epithelial cells, and then you have the epithelial cells that bend upward as this follicle grows, and you get this coordinated morphogenesis that occurs. So this is what we're looking at in section D here. Now in section A, we're looking at the gene expression space of avian skin dermis. So we look at if we look at these different spatial regions, we can identify the genes that are involved in these different spatial regions, and we can use a UMAP representation to represent next-gen sequencing data that describe this gene expression space. So we have this UMAP analysis where there's this point cloud, and within this point cloud you have different molecules that belong to these different spatial regions. So for example, here are the genes that are involved in margin, here are the genes that are involved in core, and if you look down here you see that the margin and the core are distinct. They have distinct functions. The margin serves to sort of as the base of this follicle. The core is the expanding part of the inner follicle. And so you can see that they have distinct gene expression profiles, meaning that they have distinct gene expression. So if you look at the core and the margin, there's a boundary. The margin cells set up a certain gradient of gene expression. The core cells set up a certain gradient of gene expression. When they intersect, you get a boundary between the margin and the core. And so you can see that even though they are distinct in gene expression space, they interact in anatomical space. B is where we have supracellular biophysical effects of morphogens. So these are different aspects of function, and then you get different types of biophysical effects. So we can see here that elasticity leads to FGF expression and BMP expression, and so you have these different properties here. This is a micropipette aspiration, so they're kind of pulling on this tissue. So when FGF is expressed, there's less aspiration, so that means elasticity is limited. So the idea is you're aspirating against the surface and you're pulling it into the pipette. And so if the material is more elastic, you get more of it in this pipette tip. And so you can see for BMP, you get more of it in the pipette tip, suggesting that BMP expressing tissues are more elastic, FGF expressing tissues are less. You get viscosity, which is where you have these spheroids merging together. And when you get a lot of BMP expression, there's merged. And when there's FGF expression, there's unmerged. And then activity, which is collagen disc contraction, you get more disc contraction for BMP versus FGF. So you can see the size of the disc changes. Now the color coding represents the core and the margin. So BMP is expressed in the margin, which is the blue, and the blue or the margin cells have these different properties. So you can not only look at the at these type of cells as having different spatial collocation and expressing different genes. But if you look at a single factor, which is one of the main factors that are expressed in morphogenesis, FGF or BMP, they have different property. They provide different properties to these tissues, different physical properties and functional properties. Then C, we have our parameter space of supracellular phases. So we have activity versus elasticity versus viscosity. So we're looking at activity as this collagen disc contraction versus viscosity versus elasticity. So that's what we're plotting in this three-dimensional space, these three properties. And then we see that if we take a control tissue, it's very low in activity. It's moderate in elasticity and viscosity. This FGF core, which is the core here, which is solid-like, it has very little elasticity and viscosity and very little low activity. Or I guess, yeah. So, and then in the BMP expressing margin, which is this blue area, it has very high viscosity, middling elasticity, and high activity. So you can see that the BMP margin is fluid-like and contractile, so the physical properties, move this around in parameter space. These two tissues are different. There's this gradient that's formed for each one. There's this boundary, and they have different gene expression profiles. So there's this tissue differentiation, which then has to sort of play a role in tissue morphogenesis. So let me read the conclusion, and then we'll move to the paper. So I'll start up here, where they talk about their hypothesis. We hypothesize that the emergence of these two materially distinct adjacent tissue phases produces an instability whereby the active contractile fluid margin propels the inner solidified core into the epidermis to induce budding. So there's this instability that forms between the margin and the core. This margin, because of the differential behavior of these two tissue types, the margin propels the core up into this layer of epidermal cells. It bulges upward, and then that forms this structure. So these are two material distinct adjacent tissue phases. The inner solidified core is shot up into the epidermis, and it induces budding. Indeed, by coupling quantitative phase field modeling and experimental data, we showed that these two phases create an unstable complex resolved through budding. So this is, when these two things are in sort of, first of all, they're different types of tissue. They're expressing different types of genes. You get these gradients that, form a boundary. Then they're unstable sitting next to one another. So they need to sort of resolve the tension, and you end up with this budding. So this is an interesting way to kind of look at this process. So our work shows that understanding the role of morphogens in morphogenesis requires characterizing emergent material and mechanical properties at the supracellular scale. So this is where we're looking beyond the cellular scale to these tissues and to these tissue-wide length scales. This stands in contrast to the prevailing view that functional effects of morphogens are on properties that can be characterized at the scale of individual cells, such as concentration sensing, proliferation, and chemotaxis. So there's this idea that, we can really only understand the functional effects of morphogens on single cells instead of tissues. And this is showing that cells acting collectively in these tissue types actually can do this as well. So, we're not really worried here about chemotaxis or some sort of cellular sensing. We're thinking about the collective behavior of cells. This paradigm highlights the need to distinguish between the proximal effects of morphogens, which include modulating gene expression of individual cells, and their ultimate functional effects, which enable the formation of distinct supracellular phases that are capable of morphological transformation. So again, there's this supracellular phase, which is sort of the tissue level of organization. And this is kind of what we're dealing with here. So it's not just individual cells that are doing all the work here in terms of responding to morphogens and producing it. We think of collective behavior, we think of single cells doing things collectively, and then we kind of wave our hands and say, this is emergence, or then a miracle occurs, or something like this. What we're saying here is that cells are in this tissue. The properties of the cells are just fundamentally different, and there's interactions between these two cell types, not just in terms of gradient boundaries, but in terms of like resolving the distinct modes of behavior between these two tissues. So the full paper is here. This is a science paper. Morphogens enable interacting supracellular phases that generate organ architecture. And so this is where we get into the, sort of generating these bigger length scales of organ architecture. And the idea that morphogens in the form of BMP and FGF, in the form of BMP and FGF, can regulate those. So the editor's summary: morphogens are secreted molecules that have critical roles in the generation of tissue morphology, but how individual cells affect scale up to create supracellular structures composed of hundreds to thousands of cells remains elusive. Focusing on avian skin, Yang et al. Discovered that the two morphogens enable mechanical properties that manifest at the supracellular level. So this is another perspective article, Banavar Nelson, which can look into if you're interested. Functional effects of morphogens in large tissues can thus be understood by consideration of emergent supracellular properties. And again, this goes back to the hypernetworks, because we can actually define in our hypernetworks these categories that are bigger than the individual cells. And we in the examples I gave, they're kind of like very simple, where you have a tissue type like neuronal tissue. And I had several categories for neuronal tissue. And they were not really clear in the example. But if you think about this in terms of coming up with a framework for this, those kind of different categories could be different parts of that organ, or different parts of, different constituent cells that have different properties. They may express different types of have different types of molecular signatures. As I said, the components of a hypernode can be molecular components forming a network, or it could be different cells that have different, varying properties. If you think about these different categories as having different properties of different molecular profiles and different functional properties, then we can begin to see how we can represent something like this. So we can get to that supracellular level. And that's an important part of development, as we see in this paper. So this kind of goes through some of what we saw in the preview. This is the figure that we went over. I don't have the full article up, but this is just showing the full article, sort of the name of it and the citation. Okay. The next paper I'd like to talk about is this optimal control of gene regulatory networks for morphogen-driven tissue patterning. And this is by Albert Pezzotta and James Briscoe. And so, again, we'll go over the graphical abstract in the in brief. So the in brief reads: an approach based on optical control theory using reinforcement learning to identify signaling dynamics that guide cells to specific fates in a timely, precise, and reproducible manner, offers insights into how morphogen-controlled gene regulatory networks pattern tissues and establish an alternate framework to the French flag model. So they'll talk about the French flag model, which is where you have these stripes that look like the French flag. I'll talk about it in the paper a little bit. So this is using a lot of mathematical tools as opposed to experimental tools, although they do use some experimentation here. They're using optimal control theory, reinforcement learning, and they're looking at signaling dynamics. So they're actually looking at cells here, but they're also looking at these kind of tissue-wide patterns. So this shows the regulatory landscapes and the morphogen-driven patterning. So here we have regulatory landscapes. We have a landscape, which is usually one of these dynamical network landscapes where you have bistability. So this is the bistable point. And a cell on this point can either go in one direction or another. These valleys represent distinct states that cells have, that they can change their state if they can climb out of this energy well and come up to the bistable point, at which point they can transition into another state. But, they've used these landscapes to represent sort of the way that states are sort of evolve over time. So we have a cell that's, usually stem cells are poised at one of these bistable states to go into a number of states. Ultimately, they'll probably go into a single state and stay there, but sometimes they'll pop out and go into another state. Like, for example, a cell that goes through apoptosis or a cancer phenotype or something like that. So these energy landscapes are, we use the parameter V, which is, usually associated with some sort of physics, representation or physical parameter. But it's really just a conceptual model to show that, you have these distinct states, that they're sort of valleys of potentials. In other words, once you're in a state, it's hard to climb back out. You have to put a lot of effort into getting out of that state and moving to another state. Whereas if you're poised at a multi-stable point, it's easy to go into one state or another, given different things going on in that cell. So we have this representation here of these two cells where there's some sort of, relationship between this one cell state and another, and there's either a brake or an accelerator put on that cell. And this says optimize speed, accuracy, and energy trade-offs. So there are these different trade-offs in differentiation. So there's this uncontrolled mechanism here versus a closed loop mechanism. So basically in the uncontrolled mechanism, we have this feed forward that drives it from one state to another. You have this feed forward of a brake or an accelerator. In the closed loop case, however, you can optimize the speed, accuracy, and energy trade-off set of trade-offs that need to be made by introducing this parameter phi. And so this is the control. This is where they're pulling in reinforcement learning and optimal control theory. So you can actually shape this process with some external parameter. And there's this closed loop feedback that forms between the initial state and the brake accelerator and then the final state. So you can actually shape where that goes. So you see here, it's no longer a bistable state. You're just moving towards a final state. And there's this control mechanism that makes sure it gets there. And so then this is the equation that we use to define phi, which is negative change in V control. There's this gradient that's defined as a negative gradient going downward. That's your V control, and then VF, which is effective, which is V plus V control. So basically you're adding the control component to this process V, which is, you see here, is the landscape. So landscape V going negative, and you have this control process that guides it down into the well. And so you can see from your initial state to your target state, you gradually get to your target state. So that's a regulatory landscape. Now we talk about morphogen-driven patterning, which is where we have this closed loop control. We take this phi parameter, we plug it into this process, so the regulatory landscape is a single cell. These single cells individually go through this process of finding its state. It can either be uncontrolled, which is an open loop feed-forward system where you have this bistable state, and then you go into one stable state or another, or you can have this closed loop control where you have this phi parameter, which basically guides it down to a state. But in morphogenetic driven patterning, you do this in multiple cells at once, or you do this on a gene regulatory network. So there's this closed loop control parameter phi. It controls this process at every step. So the morphogen is actually sending a signal to both transduction and to this closed loop control system. Transduction is also sending a signal to the closed loop controller. These two processes, morphogen and transduction, lead to an effector and a target genetic regulatory network. So this is where we have morphogens acting on a cell, things are being transduced into the cell. Then there's an effector, which is the behavior of the cell. And then there's this target or into the gene expression mechanism. And then you have this target genetic regulatory network where you have these genes operating or cells sometimes operating in a network. And so closed loop control then takes a signal from the GRN as well, and then sends a signal to transduction and effectors to control this process. And so you can see that morphogen dynamics change. You get this gradient, and you go from morphogen dynamics and a gradient to patterning dynamics, where you get this gradient over developmental time. So that's what they're talking. This is very different from the French flag model. So the highlights of this is morphogen signaling controls the pattern of gene expression in developing tissues. Optimal control theory identified signaling mechanisms for morphogen patterning. By incorporating feedback between signaling and gene regulation, it explains dynamics. It, being the model, provides an alternate framework to the French flag model for morphogen patterning. So this French flag model is the stripe model where you have these cells that are interacting in these gradients, and the boundaries between gradients define the edges of the stripes, and you get these distinct stripes. And so this is a different model which uses this sort of closed loop control. So the summary reads, the generation of distinct cell types in developing tissues depend on establishing spatial patterns of gene expression. So this is the French flag model, where we have these stripes that are organized in space, one after the other, and they're in parallel. Often this is directed by spatially graded chemical signals, known as morphogens. So there's a gradient that happens, and then there are boundaries between the gradients. You get boundaries, and then that's how you define and the cells within those regions are defined by whatever those gradients look like and the boundaries look like. In the French flag model, morphogen concentration instructs cells to acquire specific fates. So depending on what part of the gradient you're in, which gradient you're in, this tells the cell, this is your fate. So if you're in the left-hand part of the French flag model, you'll acquire a certain fate. If you're in the right-hand part of the French flag model, you'll acquire another fate. It's really depends on, what kinds of things the chemical milieu is guiding this process of differentiation, but it's all spatial. And so, it kind of almost seems like a tautology. You say, well, okay, there's this gradient, there are these boundaries, the cells within these parts of the gradient are reinforced, and you get this pattern formation out of nothing. But then the question is, well, how does it begin? Is it just that the cells start expressing these factors autonomously, or are they responding to signals in the environment? And if they are the signals patterned in the same way that this French flag will form, we don't, that's stuff that people kind of tried to work out, but really I think there's some assumptions there that are maybe tautological. So this is kind of where they're coming from on this. How this mechanism produces timely and organized cell fate decisions, despite the presence of changing morphogen levels, molecular noise, and individual variability, is unclear. So we don't necessarily know the origins of this. In addition, there are a lot of things like there are fluctuations in morphogen levels, there's molecular noise, there's individual variability, which means that cells respond differently to different types of signals. And so those should not necessarily result in a flag with, solid colors from left to right with stripes, but it should lead to some sort of mosaic nature. So you could have stripes, but the stripes would be mosaic. Or there might be, cells kind of drifting across the boundaries. We don't really know, we have to account for all those things. So this is something that we really need to think about in terms of feedback. We kind of think of cell differentiation as this feed-forward process, as I said, but you also have feedback. Sometimes that feedback can be kind of like, adding more of something to something, but sometimes it's control. And so that's what they're trying with their feed parameter here. They're adding a control mechanism that monitors what's going on in different processes, and then using that to sort of guide the process of differentiation. So this accounts for noise. You can plug noise into the phi term. You can plug individual variability into the phi term, and so forth. So moreover, feedback is present at various levels in developing tissues, breaking the link between morphogen concentration, signaling activity, and position. So that this feedback, and if you think about this at various levels of tissues. So if we go back to our figure, the regulatory landscapes of individual cells, in the morphogen-driven patterning, we see that we're dealing with two different scales of organization. We're dealing with single cells, and oftentimes the single cells are doing this whole process of finding things on the landscape on their own, sort of like independently. But that's, of course, not the way biology works. Cells are in, proximity to each other. Cells are not in proximity to each other. And so that has to play a role, you would think, in some of these regulatory landscapes, that they may look different from one end of the French flag to the other. Furthermore, we have this morphogen-driven patterning, which involves multiple cells. We have GRNs, which are different in each cell. But we have to account for that. So we need this sort of closed loop control that provides feedback to the different cells, and the GRN is impacted in different ways depending on its structure and depending on its activity level and so forth. So this kind of model that they're introducing here can deal with these things and kind of breaking this explicit link, but also sort of, providing us with a means to have some sort of coherent pattern formation at the supracellular level. So here we develop an alternate framework using optimal control theory to tackle the problem of morphogen-driven patterning. So to recap, intracellular signaling is derived as the control strategy. So intracellular signaling, which is within the cell, you have signals that are kind of feedback from the environment, feedback from what the cell's current state is. This is derived as the control strategy that guides cells to the correct fate while minimizing a combination of signaling levels and time. So the signaling levels are kind of sort of averaged over, or they're given a sort of a summary by the feedback mechanism. And again, we don't know exactly what the feedback mechanism is in terms of biology, but we know that cells do. People have proposed things in systems biology, such as cellular signal processing, where signals are summed, or there's some sort of signal processing going on with respect to smoothing or peak clipping or things like that. So and these like morphogens are all analogies. So we have to take that with a grain of salt. And so this is basically the control strategy, intracellular signaling. And of course, intracellular signaling is incredibly complex. So it's easy to throw your fate in with a very complex system and say, well, whatever comes out the other end will be the answer. So that's another grain of salt. This approach recovers experimentally observed properties and patterning strategies and offers insight into design principles that produce timely, precise, and reproducible morphogen patterning. So that's they've actually tested this out, and it's something that I think is probably it's a candidate. It looks like it's a workable model. I don't have any real problems with it. So one of the key things here are these gene regulatory networks, or GRNs, in the responding cells. So these GRNs comprise of intracellular signaling pathways, and these are where we have more, interactions between morphogens, things that control the expression of morphogens in single cells. So you're actually getting down to the GRN level, which is where the morphogens are actually being produced. And the downstream transcriptional responses, which are central to transforming the continuous spatiotemporal input of morphogen signaling into discrete cell fates. Again, with GRNs, you're defining a network of genes. You're saying this is, the way this is what the cell is doing. But, again, you have to pick the things that you're interested in for your GRN. So that's a representation of this intracellular signal. It's a stand-in for things that may or may not be sort of the primary drivers of this process. We can say that, GRN of morphogens is the answer, but, I don't really have a problem with doing it that way, but it's a choice. Regulatory interactions between components of these networks appear to perform the equivalent of an analog to digital conversion. So this is what I was talking about with cellular signal processing, where these regulatory interactions perform this analog to digital conversion. In other words, these analog signals, you have morphogens that occur at a certain concentration. That concentration is analog in nature because it goes up and down with the production of the activity of the gene regulatory network and the production of morphogen. And so that is, analog. That fluctuates in an analog manner, but then it needs to be converted to some digital representation or digital thing that the gene regulatory network can then act on. So whether that be a threshold, whether it's above or below a threshold, or something else like that, where it's a number of discrete states, that's the kind of thing that they're proposing that these regulatory interactions are doing. So, moreover, nonlinearities in feedback within the GRN can confer multi-stability, memory, and hysteresis to cellular decision making. And again, we have that example of multi-stability where, a cell can sit in a multi-stable state and go into one fate or another with ease. So you could have a cell that could be poised to go into 2 or 3 states, depending on other things going on in the environment. And it wouldn't be that much of a task. It's just kind of like where you're poised to, go into one or another state. So, a lot of stem cells or precursor cells are like that, where you have, like, if you're a neural precursor cell, you can go into a number of neural states, but you're not going to become like a bone cell or a muscle cell. Other cells could become either a bone or a muscle cell. It just really depends on the overall state of the cell. And so this is something that's an important property of this kind of a model of morphogenesis. Then, of course, memory and hysteresis. Hysteresis being the inability to return to a previous state. So, if you want to lock a cell into its state, you want hysteresis, where it can't just move back to being a stem cell or move to another state easily. And so these are things, this is this whole literature on cellular decision making. Again, this is an analogy to human decision making or animal decision making, which is not really the same thing. It's just kind of referring to how changes happen in a cell and how they're sort of initiated. So this is a different kind of, they use the term decision making, but it's not at all like what we would see in, say, the brain. A consequence is that cell fate depends not only on the levels of signals and effectors, but also on their temporal features. So again, as they evolve over time through development, as they make these decisions, as they change their fate, the temporal features are also kind of feeding back on this. So feedback allows you to take those into account as well. Taken together, the complexity of interactions within the GRN can produce rich dynamics in the signaling and gene expression of developing tissues. Understanding the origin and function of these dynamics offers insights into patterning. Moreover, the interplay between morphogen gradient and GRN allows cells to actively contribute to morphogen signaling, rather than simply being instructed by the gradient. So it's not a top-down process, it's an emergent process, but it's emergent through individual cells doing their thing, and then there being this feedback mechanism, and then interacting with other cells. So this is where we get this gradient is sort of a top-down, sort of a template that we assume because we see this pattern. But that's, of course, not necessarily the way it works. We need this bottom-up emergent phenomena that we, that's kind of what we had initially started out with, and then we impose something from the top down, some kind of cheat, using the French flag model. This highlights a need for alternative paradigms to the French flag model, in which the GRN plays a complementary and equally important role to the morphogen to frame questions about morphogen activity. So we can look at the dorsal ventral patterning of the developmental vertebrate neural tube. This is a well-established example of a morphogen pattern tissue. And so this involves sonic hedgehog being produced from the notochord and the floor plate, which are located at the ventral pole, which is at the top, forms a ventral to dorsal gradient, so you get less sonic hedgehog as you move down. And so this involves a number of other molecules. So you see that these morphogens can be, pretty complex. And so if you have a network of them, then it makes it easier to account for these interactions. So the neural tube GRN, combines sonic hedgehog with the Glee family of proteins. So this includes members of the NKX, OLEG, PAX, and ERX families. So you can include all of those in your GRN. Then dynamical systems theory provides a framework to describe the activities of morphogens and GRNs. As we saw, we had our model of our potential model with the wells, and we had V as the parameter that described the sort of the energy, negative energy minima. So like a negative V is downward, a positive V is upward. And that's from dynamical systems theory. And that's a good way to describe it from a physics perspective. But then we have to map that V to something in the cell, something biological. And that's kind of where it gets a little tricky. But, basically people think of this as like potential or cell state or something like that. So the behavior produced by such models can often be represented geometrically as a dynamic landscape. This provides an intuitive description of cell fate decisions that correspond to the idea of an epigenetic landscape proposed by Waddington. So this is actually very similar to the epigenetic landscape, but it's not identical to the epigenetic landscape. So this is a landscape that is sort of describing cells. They're using a model from physics, and Waddington's model involved these channels or canalization, where these wells are actually parts of channels that move in time. So they're moving, this sort of bistable or multi-stable state is really the top of like the channel or the canal, and then the canal branches into two, and you can take one route or another down. So you're always minimizing V in the epigenetic landscape. This doesn't really form an epigenetic landscape, however, in the conventional sense. This forms kind of like more of a, something that always leads down to one single channel. So, this is kind of a different view of this. I don't know how this would map to an epigenetic landscape versus the uncontrolled, but they do mention that this kind of ties into epigenetic landscapes since Waddington. So in this view, the developmental trajectory of a cell is analogous to a particle rolling on an undulating landscape where we have these valleys or these wells and watersheds representing fates and decision points respectively. Morphogens can be thought of as tilting the landscape in such a way that the valleys can be made deeper, shallower, disappear altogether. So those wells that we saw, represented by negative V, they can get shallower or deeper. So that negative V can be larger or smaller, or the maximum negative V can be larger or smaller, depending on the morphogen activity. So this is an interesting kind of insight because, morphogens are really controlling this process. In this way, the morphogen controls the terrain and hence the valley a cell enters. So again, if we map this back to these energy wells, those energy wells are entered as we get some sort of decision-making happening by the cell. So nevertheless, it remains a challenge to construct landscape models that incorporate the knowledge of signals and GRNs. How is the landscape modified by an external signal, and how are feedback mechanisms incorporated? How can experimental inferred landscapes give insights into the signaling dynamics? So these are things we don't, these are things that they tried to answer in this paper. This actually talks about Mars levels. This framework, made explicit for the visual system, has become a widely accepted paradigm for understanding general cognitive processes and information processing in biology more generally. The distinct levels pertain to the tasks performed. Problems solved by the system under consideration, which is the computational level, the rules and representations used in solving these tasks, which can be described in input-output mathematical laws or the algorithmic level, and the physical realization of these laws, the hardware and physical processes used to solve the computational problem, or the implementation level. So they got to actually break this problem down into more levels, which I find interesting. So this is the reinforcement learning solution for the optimal control of ventral neural tube. So they're using ventral neural tube and the GRN associated with that to do the simulation. So this is where they're doing this reinforcement learning solution. So schematic representation of the closed loop control for the system. The activator and repressor forms of Gli protein are given by the function of the neural progenitor markers PAX6, OLIG2, NKX 2.2, and ERX3, which evolve according to the stochastic dynamics defined in STAR methods, which are in the paper, and ventral neural progenitor GRN model, which is this PONI network. So this is kind of an example of the expression, which is in arbitrary units, and time, which is in arbitrary units. And so we have these. This is the GRN here, where we have Gli-A, Gli-R, PAX6, ERX3, OLIG2, and NKX 2.2, and their respective colors. And then you see the differential equation here, and then this other equation with phi in it. Now, this is the expression here. So this is a profile for different genes in the network. This is Gli-A. This is OLIG2, PAX6, ERX3. And then this is Gli-A versus Gli-R in this figure here. And then this is OLIG2, ERX3, and PAX6 in the bottom. So you can see the dynamics as they fluctuate. So basically, in these GRNs, you get these fluctuations of activity over time. Some of them settle down and some of them rise in terms of expression. So NKX 2.2 rises over time and then hits a plateau. OLIG2 has a big pulse at near the beginning of the process and then dies off. Gli-A sort of establishes things, so does ERX3. They kind of have this initial high activity, and then falls with Gli-A exhibiting a small peak early on. And so you can see this, as you run these models and the interactions between them, you see different patterns here. So this is a reinforcement learning solution. The optimal control problem is solved separately for two different targets, A and C. The NKX 2.2 plus target, D and E. And so these different figures here show, let's see. So if you go up to A, this shows the network B, C, D, and E. We'll focus on B and D here. D and E are the OLIG2 target. A and C is the NKX 2.2 target. And so this kind of goes through B is a sample of the controlled dynamics for the NKX 2.2 plus target. Solid lines are the medians, and shaded areas are the 10th and 90th percentiles. So you see that it's, number of runs, and you see the shaded area, which is like the variation in the runs. The control U star, comprising activator and repressor Gli top, and the gene expression dynamics bottom. In the solution, although the repressor Gli remains always low, the activator Gli exhibits an initial transient characterized by high values, and then reduce drastically at steady state, reminiscent of the adaptive dynamics observed in experiments. So they're actually replicating what they see in experiments here. You get these different phases, these transient bursts of activity, and then these plateaus where things are stable. So this is basically their simulation results. And so you can also apply reinforcement learning to morphogen-driven patterning tasks. You have a number of results here. And then we have our methods, which are available online here. So these are detailed methods, which we won't go through right now, but they're, different types of things for the reinforcement learning solutions, multi-agent control dynamics, environment dynamics, optimal control and potential, and their optimal stochastic control and solution methods. So this is pretty informative if you're interested in this kind of modeling. I'll read the last paragraph to finish here. The optimal control approach, with its focus on linking mechanisms with control, is ideally suited for the analysis of in vitro and synthetic systems. This could be used to design and refine signaling regimes from the directed differentiation of stem cells in vitro and the production of specific sets of cell types in defined proportions. An understanding of the control principles operating in biological systems may provide insights and inspiration from the construction of artificial systems, as well as support the use of stem cells in disease modeling and regenerative medicine. And with that, thank you for paying attention, and I hope you learned something.